Hello and welcome to the session of the sodium. Sodium is one of the most important electrolytes. Let's see the function of the sodium. It helps cells to uptake nutrients and water. It helps muscles to contract. It helps nerves to carry messages between brain and the body. It helps balance body's fluids and regulate blood pressure. Let's see some important points about dietary sodium. The high sodium intake leads to increase in the extracellular fluid volume and hypertension. 0.5 to 1 gram of the salt per kg body weight, which is a huge amount of sodium, which may lead to fatal outcome. Food naturally low in the sodiums are fruit, vegetable, oils and cereals. They contain up to 20 mg of sodium per 100 gram. The next is meat and fishery products. They contain 40 to 120 mg of sodium per 100 grams. Some shellfish and mussels and oysters contain 500 mg of sodium per 100 grams. Whole milk contained 50 mg of sodium per 100 gram. The sodium content of the processed food obviously varies depending on the amount of the salt added during their preparation. Frozen foods contain up to 700 mg of sodium per 100 grams. And traditional meats and cheese contain very large amount of the sodium up to 2500 mg of sodium per 100 grams. Let's see the relation of the sodium to table salt. The sodium chloride or the table salt, it is approximately 40% of the sodium. 1/4 teaspoon of the salt is equal to 575 mg sodium. 1/2 teaspoon salt is equal to 1150 mg of sodium. 3/4 teaspoon salt is equal to 1725 mg of sodium, and 1 teaspoon salt is equal to 2300 mg of sodium. The American Heart Association recommends daily intake of the sodium must be less than 2300 mg per day, and it is recommended to have a sodium less than 1500 mg per day with the patient suffering from high blood pressure. WHO guidelines sets the limit of the sodium intake for less than 2000 mg per day as a target. Sodium concentration in our body is maintained in the narrow range of 135 to 145 milliequimole per liter of the plasma. The value is 145 to 155 milliequimole per liter of the plasma water. The sodium is a major cation of the extracellular fluid. Active energy requiring processes at the cell boundary exclude the sodium from the intracellular compartment and confine it to the extracellular fluid. Two mechanisms serves to maintain the plasma sodium concentration in our body in the narrow range in a healthy subject is first is a thirst and the second is a vasopressin ADH hormone. Let's learn osmolality versus osmolarity. Osmolality is an estimation of the osmolar concentration of the plasma and is proportional to the number of the particles per kilogram of the solvent, and it is expressed as milliosmol per kg. Osmolarity is an estimation of the osmolar concentration of the plasma and is proportional to the number of the particles per liter solution, and it is expressed as millimole per liter. Plasma osmolarity. The contribution of the sodium to plasma osmolarity is apparent from the formula below. Plasma osmolarity is equal to 2 into sodium plus glucose upon 18 plus blood urea nitrogen upon 2.8. Sodium unit is milliequimole per liter, glucose unit is mg per dl, BUN unit is mg per dl, and osmolarity reference range means normal range is 275 to 295 milliosmol per liter. Sample collection, storage and handling procedure. The serum sample, SST, plain tube, lithium, heparin, plasma and urine. These are the samples required to conduct the sodium test. Separated serum or the plasma should not remain at 15 degree to 30 degree centigrade longer than 8 hours. If the assays are not completed within 8 hours, the serum or the plasma should be stored at 2 to 8 degrees centigrade. If assays are not completed within 48 hours or the separated sample is to be stored beyond 48 hours, sample should be frozen at -15 to -20 degrees centigrade. Fasting is not required for the sodium testing. Interference. No interference from the bilirubin less than 4 plus or lipemia or hemolysis. The sample with plus 4 lipemia performs sodium on the lipoclear treated serum. Methods. There are different types of the method that are used in the clinical laboratory to detect the sodium. First, we learn about the ion selective electrode. The ion selective electrode makes the use of unique properties of certain membrane material to develop an electrical potential, which is also called the EMF, electromotive force, for the measurement of the ion in the particular solution. The electrode has a selective membrane. This is the selective membrane. The selective membrane in contact with both the test solution and the internal filling solution. The internal filling solution contains the test ion at a fixed concentration. The concentration we know. Because of the particular nature of the membrane, the test ion will closely associate with membrane on the each side, and the membrane EMF, electromotive force, is determined by the difference in the concentration of the test ion in the test solution and the internal filling solution. So this is how the ion selective electrode works. The next is enzymatic method. The method is a sodium activated enzymatic reaction between the sodium dependent BD galactosidase and its substrate ONPG, converting it to O-nitrophenyl and galactose. The rate of the product formation as a 405 nanometer corresponds to the amount of the concentration of the sodium present in the sample. So ONPG is treated by sodium and BD galactosidase, which converts into ONP and galactose. This reaction is measured as a 405 nanometer and it corresponds to the amount of the concentration of the sodium present in the sample. Let's learn the next method. It is a colorimetric semi-automated method. The method is based on the reaction of the sodium with a selective chromogen producing a chromophore whose absorbance is directly proportional to the sodium concentration in the sample. This is the sample of the testing kit literature available for the sodium. It is showing how to perform the assay. The reaction type of this type of the test is endpoint reaction. The wavelength where it is measured is 630 nanometer. These are the reagent and the sample volume described here. Assay procedure. The pipette into the clean dry test tube labeled as a blank standard and the test. And add sodium reagent standard and sample according to this table. Mix well and incubate at the room temperature for 5 minutes. Measure the absorbance of the standard and the test against the reagent blank at a 630 nanometer. So this gives the concentration of the sodium. The next is flame photometry method. Flame photometry is one of the old direct potentiometric method commonly used to measure the concentration of the sodium, potassium, lithium in the serum and the urine sample. It is routinely used as a reference method, working on the emission photometric principle. The flame photometric principle is based on the chemical property of alkaline earth metal. On the high temperature, absorb the energy from the heat source being conducted to a state of the excitation in their atomic form. When the return of the initial energy state, the radiation is released at a specific wavelength, being some in the region of the visible spectrum. Thus, the emission is proportional to the concentration of the ion present in the study sample. If an appropriate photodetector is placed behind the filter, an electrical signal is obtained being proportional to the concentration of element in the solution. Important points from these videos are: restrict dietary intake of the sodium up to 2 gram per day for the normal adults and less than 1.5 gram per day for the hypertensive patients. The reference range of the sodium in the serum is 135 to 145 milliequimole per liter. These are the references for this video. Hope you like it. Thank you and see you in the next video.